ago. Even 5, 10 years ago, it was really just too expensive. But now you can do it really quickly. There's even a discussion that maybe it'll get cheaper, and this is, it's moving faster than Moore's law, which is doubling the computer power of every, sort of computer every 18 to 24 months. That's why your phone is more powerful than supercomputers from the 80s, and really highlights, that power. But it's even faster than that. So this is the fastest technological change that's ever occurred in human history. And there's rumors that maybe the genome will get down to $100 kind of soon, which means that it's getting easier to make more data every day. So every day that you wake up, there's more data today than any other day that's ever existed in genetics. And so as a geneticist, that means that every single day that I wake up is the best day to be a geneticist. It doesn't mean it's the best day to, you could have a bad day, like you could fall down the stairs, or you could, like, step on a grape in the kitchen or something weird that doesn't feel pleasant on your feet, feels like it's like an eyeball. That's kind of gross. But it means in terms of just the best day that you can possibly have in terms of capacity for discovery and analysis of data, it is empirically the best day ever. And so that's the human side. We're thinking about how many human genomes there are and how much genetic data there is. But of course, there's more than one genome in the world beyond just the human genome. If you could see the world through the microbiome, it would look something like this. So basically, there's aerosolized droplets, which I think we've all been thinking a lot about more recently because of COVID. But normally they're innocuous and floating around you. And this is microbes you can see colored here that are basically on the surfaces that you touch every time you sort of move an object. If you think about sort of what's on your skin or on your hand, think about looking at what happened if you kind of imagine your skin as its microbiome, it would kind of be illuminated here. Think about what you're breathing out. So there's aerosolized droplets that can contain microbes. Think about what's inside your body. You can see in here the gastrointestinal tract is lit up in there, and you can see the chair, the keyboard, the mouse, the speaker, really everything in a room, anywhere you are, has its full microbiome sort of ecosystem. And this is also true like in a cup. If it's got microbes in the bottom, when you throw a pen or pencil in a cup, you get this poof of microbes that can happen. So really, everywhere you go. Some people find this video absolutely disgusting. I think this video is absolutely fantastic and interesting because it's actually true. Even the buildings in which we live and work have their own, what we call built environment microbiome. You can see is illuminated here. And these microbes are everywhere, but if you compare them to human cells, they're pretty small. Like the human cells are big, eukaryotic cells, bacteria are small, prokaryotes, and there's some yeast are eukaryotes, but you can see they're pretty small, but they make up a fair amount of your mass. If you're, say, maybe a 200-pound human, several pounds of your body is actually this extra organ of the microbiome. It's actually kind of like, think about your kidney, think about your liver, think about your microbiome the same way. And not just because it's there, but it's also biochemically active. And so estimates from Lee Hood, who we recently been working with on another project called Longevity, he's said that estimated about a third of the molecules in your blood are either made by the microbiome or modified by it. So it really is this little miniature pharmacy inside your gut. If you're looking for the nearest pharmacy, just look down by your belly button. And this has led to a greater appreciation in the past 5, 10 years that this little pharmacy inside of all of us, is really linked to all sorts of things that change in your body. And so really large projects like the Human Microbiome Project or international consortia that are looking at this have linked it to obesity and how you process food and inflammatory bowel disease. And even, all these diseases that previously were human diseases that made sense, like ulcers, or you think of acne, or even obesity could be contributing, but things like depression or autism, or thinking more about the neuropsychiatric disorders, were kind of odd to think about as really being impacted by the microbiome, and even really controversial to say 20 years ago, are now the subject of many, many large research projects. And so this is because we keep finding that they're relevant in all kinds of areas, including cancer. For example, you can find intratumor bacteria that defend the tumor against basically chemotherapy, that you basically have to first do antibiotics to get rid of the bacteria in the tumor, and then do the chemotherapy to treat the cancer. So really surprising and unusual connections between the microbiome and cancer. There's been other papers that show that if you take drugs that are targeting like human cells, about a quarter of them will actually change what happens to the bacteria in your gut, at least in vitro studies. And so it seems that, you think you're taking a drug for your cells, it's actually for all your cells in your body, including microbial. And so that's kind of interesting. And, really, you think about who the drug is really for. And the other thing is, think about the organisms that are in you and on you. Essentially, if you look at what they're making, or what are called biosynthetic gene clusters, you can take normal flora from, say, normal skin microbiome or vaginal or gut microbiome, and just look at the species and say, okay, well, can we find a new species and build, these are called biosynthetic gene clusters, or components of genes that come in a cluster that have one biosynthetic pathway and can make little molecules. You can predict it from the sequence data alone. And this has been used to predict new antibiotics in the literature, and also been followed up by many different companies. And so what this means is, given that backdrop, and I could go on for hours about all the different microbiome studies that have come out in the past 10 years, But what that means functionally is when you think about, okay, I want to be a scientist. In particular, I want to be a human geneticist, which is how I was originally trained at Yale to medical school. And I began to have a much greater appreciation because I can't actually be a good human geneticist if I just look at human DNA. Actually, I can't do that at all. You're not a good human geneticist if you just look at human DNA. What that means is I have to be really a cross-kingdom geneticist just to be a human geneticist. And so this means that, it's helpful for human physiology, human clinical work, but understanding microbes expands to many, many other places. In particular, you can look at strange places like we have a project called the Extreme Microbiome Project, and reminds me, Krista, I got to catch up with you on the website soon, but a bunch of updates for it. But we've been using this as a place to go to really survey areas around the Earth and find, do we observe, peculiarities? So, for example, this is a candy pink lake in Australia called Lake Hillier, and here you step into the water, you kind of slosh around, get it to your feet, your ankles will start to hurt because any of your skin that's exposed to the water, the cells start to lyse because it's a hypertonic, hypersaline solution. So it actually burns your skin just because the cells are dying. It's so salty. But some other organisms don't mind the huge amount of salt that are in the water on the shore, and even can survive just fine in there, and even have this pink color. So we want to study how they adapt, how they survive. It's almost like building a genetic toolbox, and then we could then better understand how they survive at all from a real clear genetics perspective. They also have been looking at places, this is with Scott Ty in Antarctica, and examining what we see here are helicopters, or for example, you can do sequencing right in the middle of Antarctica. We've also done work in New York City in the Gowanus Canal, which is a Superfund site, and been sequencing here to look for, again, extreme adaptation, in this case to sewage and waste and toxins. And then we've also been broadening the scope. So really, we're looking all over the place, looking in places with changes in salinity, temperature, pressure, different sort of gravity. Look at the space station. I'll come back to that at the end. And also looking at other places, like for example, just looking in subways. So here you can see this strange environment that's clean and dense and full of people. Pre-pandemic, people were afraid to touch it. I would still find people with latex gloves. But I also got very curious about what's present in the subway and the microbiome. When my daughter got old enough here, who I shamelessly use in my talks. Hold on a second. Maddie, I'm using your picture in the talk. She's down the hall. She's very excited. Yeah, this is her with an ice cream cone. When she got old enough to ride the subway, she actually then, licked the subway pole on one trip. And we were really curious to know, okay, because that happened, what actually occurred during that interaction? So I became immensely curious about what that was. And so I wanted to build sort of a microbial map of a metropolium, basically the metropolis, or what the New Yorker called the metropolium. And so here, I really became curious. I wanted to see what was there. So we launched this project in 2013 to go across all 5 boroughs, swab everything, build up an app, which is now actually been updated, and also get timestamp and tagging of all the samples. And here we actually then sequence 10 billion fragments of DNA, map them to every known species at the time, and look to see how many things we could identify. And what's kind of striking, both then and now, is that of all the billions of fragments of DNA we sequenced that were in our city, half of them were from unknown organisms. They didn't match anything that was known. And you might think, that's interesting. Are there aliens in the subway? Well, no, there's not, but the databases are incomplete. It's worth noting, though, it does sometimes feel that way. If you look at subway creatures on Instagram, if you've not seen this, or on the Twitter feed, it's just a collection of weird things that you can find in the subway. I highly recommend it. Any of you have been riding in the subway, you'll see really weird things of people who just like wear bags over their head or other strange things on the subway. So I highly recommend the Subway Creatures Instagram feed if you haven't already seen it. In any case, let me go back to where we were here. So if you've not seen that, I again highly recommend it. And so might feel sometimes like there's weird creatures there, but they're not aliens. They're actually just missing species. So what we've done is also map the other species we can find across known phylogenies and figure out what they're doing. So for example, some of the samples we got from the Gowanus Canal, which looks like this, and you might think that's kind of gross. It's got sewage. Could we tell that it's sewage? We actually could. There's certain kind of bacteria called methanogens that are in there, and they're often found in sewage. And so you can pick them up right from the sequence information itself. You might think that's really gross. It's probably not safe to swim in there. There's a guy who concluded and confirmed in 2015 that it is not safe to swim in there. That's right. But what's been happening in the past few years is thinking more about what are these organisms really, like who is there and what are they making? Can we use this kind of information to really build a genetic map of these places to better understand it and think about adaptations, and also learn what's happened in a place? Basically, can the microbes themselves serve as a biosensor and biorepository of what's occurred in any given place? So for example, we saw that species diversity is different everywhere you go across New York City. You can see different species here of different species, but this one clearly stood out. It's because this was the station that was actually flooded by Hurricane Sandy and had 10 species we saw here that we didn't see anywhere else in the rest of the data set. And so if you recall, some of you might have been here for Hurricane Sandy. It came through a huge storm, knocked out some NIH websites, came through, it had huge waves that came through the city. You remember if you were in Manhattan that day, there were some waves that got really big. I don't know if any of you remember this, these really huge waves. I'm just kidding. This came from the day after tomorrow. This is not a real thing that happened. This was just from the movie. But the one map that was real is this map. Is actually, you can see here about half of the subway system was offline because some of the stations were quite literally submerged. So, for example, South Ferry Station, this was completely underwater and submerged, and we wondered if there was a cold sort of molecular echo of the ocean water that was there that we could kind of pick up. And so, when we did the sequencing and compared it to known species, we could pick up things like this Antarctic species called Chionella frigida marina that was, previously thought to be only found in cold ocean water, even near the Antarctic. And it makes something called the eicosapentaenoic acid, which is often found when you're eating fish, for example, found with the fish. And if you have low levels of this in your diet, you could even be at a risk of suicide, according to this paper. So this did answer the question of a reporter once said, okay, well, Dr. Mason, should I lick a subway pole? Is it okay to do? Tell me the answer. And I said, well, I give a very nuanced answer like a scientist would and say, well, it depends on what's there and how long it's been there and the state of the immune system of the person that's doing the licking and generally the sort of medical history of that person interacting with other people. A lot of factors, really depends. I mean, you'd probably be fine and probably okay, but it's hard to tell. And so, of course, the only thing that appeared the next day in the Gothamist was that expert says licking subway poles is probably fine. So this is my fun interaction with the media. It's a little of a lesson for everyone, but it's also important to note that this is actually true. You, in general, statistically would probably be fine. It's also important to note that you do need exposure to microbes, especially when you're young, to build up an immune system. This is something called the hygiene hypothesis, that you actually, if you lack this, this actually might have been leading to a lot of increased allergies in our society. What's called the missing microbes problem by Marty Blazer at NYU. And there's a whole great book about it, just called The Missing Microbes. And it's also been tested out in clinical trials. Like, for example, if you start to get allergies to peanuts when you're a kid, the instinct as a parent would be, well, I'll just get all the peanut butter out of the house and get rid of all peanuts and anything near them. But it's actually the worst thing to do. Is from randomized clinical trials, from this one and follow-up ones, the best thing to do is actually have low-level exposure to the thing you've beginning to get allergic to. That's the best way to avoid getting it later in life. And so this has been, again, now actually followed up by multiple trials, and it's actually an FDA approved product for treating peanut allergies. And so, again, this is important to note, but it does not apply during pandemics. Here, you don't necessarily want to lick a pole while there's COVID everywhere. But okay, so that's kind of a bit of background. So in the next sort of 10 minutes, I want to break down, okay, that's what we've done, that's where we've been, what happened, where else can we be looking, and what happens now. So we're very fortunate to get some support from the Gates Foundation as well as World Quant to launch this project in earnest in 2016. And also we've more recently gotten a larger grant from the NIH to look at also wastewater in Miami and Racine and also New York City and different places around the country. And this is all components of which now is a larger umbrella program called MetaSub, which is the metagenomics and design of subways and urban biomes. To think about, we basically want to build these metagenomic maps, which we can use forensically a little bit. I'll show you in a little bit. Build maps of antimicrobial resistance and track how it looks around the world, and also look for new biosynthetic gene clusters, or literally use this as a sequence engine to find new biochemistry, new biology. And really, we want to see, what's there, like essentially just to build out these maps even just at the basic level of curiosity. And what's extraordinary is that we've already done this for 5 years and collected over 30,000 samples. You can see here, sometimes the mayors will get involved in the sampling. This is City Councilman Ben Kales helping in New York, and teams all over the world have really been helping on this project for a long time. We have a preprint that's online and a paper. Actually, where I'm going at 5:30 is to go catch up a bit with David on the updates for this paper. But basically, you can see the preprint online here. And what this has led to is basically already from the data we've put out in sequence is a threefold increase in the size of the tree of life itself. And really almost 11,000 new viruses have been discovered across the world. Now, it's probably not a good time during the pandemic to say we found a whole bunch of more new viruses, but these are DNA viruses that are mostly phages. So they're not anything really dangerous to worry about quite yet. But what's also really exciting about these data is we picked up more than just viruses. We picked up really millions of new peptides. You can see here there's 31 million candidates down here of new proteins. And of those that are at least 20% different from any other known protein, that's actually 14%, so about 4.3 million novel peptides, never been seen before. Interestingly, if you look at the ratio of how many of them are new versus discovered per city, you can see Tokyo is number one for reasons we don't quite yet understand, but Tokyo has the most bang for the buck of discovering new peptides. The other thing we've been able to do from these data is to characterize them and then understand, well, do we see, for example, CRISPR arrays? Because, bacteria use CRISPR as their immune system to fight viruses. And can we use that to actually see if they intersect? And indeed they do. These viruses match the CRISPR arrays in the metasub data set. And there are many types of CRISPR systems. You can see the breakdown here. But you can see this is really a genetic map across all these viral contigs around the world. And what's really exciting is, we've now made this map and then made it in several ways. One of them is a global KMER index. And actually, let me put this link here and go to the website. You can see here, if you want to look for any sequence, you can type in the sequence of really any piece of DNA you want. You can type search and you'll get an index of, based on Nomad or Metasub, for example, where do we see these samples and where's the latitude and where do we find them around the world? And so it's a really, sort of exciting interface. We can align them. You can do visualization if you want to see what are the KMER builds between these different sequences, and also build all these other graphs. And so we've built out quite literally an index of everything that's ever been sequenced on Earth. And I'll come back to that in a second as to why that's important. But this is a map we can see around the world of what's been built. And what you can use this for is say, okay, well, are there species or fragments of DNA that are unique to one city or another? And the answer is, as you can probably guess, yes. We could even use this to maybe predict where you're from. You can see each of the cities here cluster a little bit differently. And we built something called a random forest classifier. It's basically a predictive algorithm to see, can I predict where you're coming from? And so here we can actually get to about almost 85%, sometimes 90% accuracy as we use more and more of our samples that have sequenced based on what species are present. And that is not as good as, say, antimicrobial resistance markers, because this is expected. These are much more mobile between species. That's what they're supposed to do, or it's what they often do. Whereas the species themselves are a little bit more stable between cities. And if you've been wondering, like, what's driving this trend, is it the population? Is it the temperature, the climate? It is very much the climate and the environment, but a lot of it's the population density of the city that makes a city very predictive. And also where it is it coastal or not? You can see here separates out how well we can predict different cities. The other thing that's notable is some cities are more predictable than others. New York City is a very cosmopolitan city. So some of these other larger cities are the ones that basically have so many people and so many kinds of, microbiome backgrounds moving through. They probably add noise to the system. So it makes it a little bit harder to tease out which ones those are. It's kind of cool actually about New York City. And this, again, all this data and papers in review and in the preprint I gave a link to before. Okay, so we're doing all this to discover essentially what's in the cities, discover new biology, new biochemistry, new viruses, new CRISPR elements. But what we've actually built is, if you think about it, a planetary genomic scale map. And so where we're using this, actually, I want to close in the last 5 minutes, is to think about what is a really exciting and novel era in planetary science, in biology and genomics, is we've reached a stage where we can think about not just what's happening on Earth, which you have these maps, but what might we see on the moon, or could we look for life on Mars, or maybe things that survive in the space station. So we've actually, been working with NASA on a variety of projects, and it's a really exciting time. It's kind of the new space race, is that NASA wants to get to the moon as fast as possible. In theory, by 2024, Kate Rubins, with whom we work a lot, might be the first woman to walk on the moon. And everyone's racing to get there. There's even cotton that has sprouted and grown on the moon a couple of years ago. And so you can't quite get your cotton khakis yet from the moon, your lunar khakis, but someday maybe you could. But we've been actually working with Jet Propulsion Laboratories, who just had a really great day, and thinking about what else could end up going, on the moon. Well, you have essentially, small lunar habitats on the moon. Or when you think about what just got sent to Mars, is the Perseverance rover. It got built in this room. You can see in Jet Propulsion Laboratories. Again, we work with Katsuri Venkateswaran as a collaborator and friend. And this robot was being built. You can see people here walking around with their bunny suits. This is what it looks like in the spacecraft assembly facility room. This is the rover before it was sort of brought down and dropped on Mars today. You can see them checking it out and building it little by little. And you can see, these people, these are the engineers, but they're humans, right? So they'll still maybe grab a selfie with the robot, because I think if I was building a robot that was going to another planet, I would probably grab a selfie. It's not necessarily crazy. But what is interesting is you can see what is not hermetically sealed. You can see little bits of their face here. Their microbiome and their human DNA gets on here, but then they clean it. They do the next layer, they clean it. They try to keep it to a very small minimum, but it's impossible to avoid, contamination. It'll never be 100% pure. And so what that means is essentially this rover, which is, just, headed off to Mars. This is what happened today, made its way through the atmosphere. landed on Mars, what it's going to be doing is actually building out these little catalogs and little containers. And the goal is to bring some of these samples back to Earth in about the year 2032. And then what we're going to do is characterize them and sequence them. And so some of the things we want to do with these samples, is not just, do some mass spec or like geochemistry, but also sequence them and see if there's any evidence of DNA. But what we have to do is compare anything we see on these samples to anything we sequenced in the clean room and really any piece of DNA that's ever been on Earth up until the year 2020 when this launched. So it's basically kind of a planetary genomic scale genetic filter that we'll be deploying for this project. And so this is just called like the Mars Sample Return Mission. You can read about it here. But it's exciting for that reason. It's kind of this idea of a planetary filter, but also if we do find anything that's on Mars, if it has any sequence similarity to anything we've ever seen on Earth, one of the first things we want to do is compare it to everything that is on Earth. And that's what you're basically building, is a complete catalog of features about every microbe and species that we've ever seen on Earth. And so that way we have a sequence-based genetic sort of association map between anything that would be found here and anything that might come from this MSR profile. So I want you to imagine looking ahead. This is a picture of the beautiful surface of Mars. You can actually see here from the high-rise photography. You can imagine yourself walking along sort of the rim of this canyon, looking down, looking out. If you look down by your feet, this is actually an image of what regolith looks like on Mars. And if you look back over your shoulder at night, you could actually see Earth in the distance here from far away. And so this is actually what the image of Earth would look like if you're standing in late spring on Mars, and that's what it might look like, and what might be, if you're maybe someone here on this call, might be one of our children or grandchildren, will actually have this view. Okay, so I want to thank everyone who made this possible. This includes, of course, NASA, the Gates Foundation of WorldQuant, and also the NIH and XC to help a lot on the compute. Thanks everyone in lab who makes this work really possible. Of course, is phenomenal and fantastic. Particularly, we've just heard from, you'll hear more from shortly, especially from Krista and Maria. And thanks everyone in the lab. This is us hanging out. It's a little bit of an old picture, but thanks, of course, to the lab, who are awesome members, and I'm very fortunate to get to work with all of them. So that's it in a nutshell. I'm happy to take any.
All right. So we're gonna talk now about what the microdirectory is. Now that you saw this great presentation about the different projects that we work in the lab that correlate with microbiome research. But the whole point of this is to actually create a database of the information about these microbes and profile those microbes. So I'm gonna start sharing my screen and give you a little bit of a background on the microdirectory. Can everyone see my screen? Looks good? Yeah? All right. So just a little bit of an introduction about myself and the team. My name is Maria. I'm here with Krista and Radwa. We work as leaders of the microdirectory. We work in the Mason Lab in different projects related to microbiome and some other related research. So we have been working with the microdirectory for about 2 years now. And we are very happy every time we receive new volunteers, because not only you are helping us, but also we feel that we are helping you to learn more about microbiology in general. And we see that the volunteers learn with a different, they finish the cycle with a different perspective of research and microbiology. So as I said, I'm going to give you a little bit of a background in the microdirectory, and then we talk in more detail about what you will be doing in the project. So as I said, the microdirectory is a project in the Mason Lab. This is a database, a collective effort to profile and annotate microbial species. Microbial species meaning groups, organisms from bacteria, archaea, viruses, fungi, and algae. We have compiled this microbial data from different databases, different studies, but we also know that there is some information that is not available there and that needs manual annotations. We give in this database a phenotypic and ecologic description of microbial species. And this is not only for our own studies, our own metagenomic studies, but also for other people that are just curious to know about a specific bacteria or a specific group of microbes. So as I said, the microbe directory emerged as an inventory of organisms in 2016, and you can find the article in Gates Open Research, and Heba, who is now a medical student, she with other colleague, she compiled like this amount of data with the help of about 30 volunteers. Then Krista and I took over the microdirectory in at the end of 2018, I'll say, and then we started getting more information to compile almost 80,000 species of microbes. You can find this information in a preprint in bioRxiv. And when we were doing this annotation, like finding information of all these microbes, we saw that, as I said, some data was stored in specific research articles. So we'll need to go one by one and write down all these characteristics of these microbes from each article. So we thought that it was going to take too long if only 2 people do it. So we decided to open the project for other volunteers to help annotating this, and at the same time learning about microbiology and microbiome research. So in 2020, more than 250 volunteers joined the microdirectory and helped us with these annotations. So how is this database structured? We know that each species has a different set of characteristics. So we decided to divide it based on each microbial group, and we have members of archaea, fungi, virus, bacteria, and algae. And for this particular case, we have divided diatoms, which are a subgroup of microalgae, but they have some particular characteristics, so we decided to group them apart. And you can see here in this diagram all the different characteristics that we're describing and we're annotating in the database about each microbial group. So for example, for bacteria, we have gram staining, if it's positive or negative, oxygen use, the metabolism. If you have a microbiology background, these terms are you are familiar with these terms, but if not, don't worry, because we have made sure that that you have all the information required to understand what we mean with gram stain, oxygen use, metabolism. And this information is stored in the CODA doc. And the CODA, which you all received a link to access this document in the acceptance email. But if you don't have it, don't worry, we'll send it again, or you can find it in the Slack channel that we're going to talk a little bit more about that in just a minute. So as I was saying, we have different characteristics for each microbial group. You can go in detail in the CODA doc, but in general, we have spores or biofilm forming, microbiome, etc. And we also have for viruses, now that we have this whole pandemic has been like a really impact. It has really impact our lives, but has been also a opportunity to learn more about viruses. And if you think about, for example, in my personal experience, my grandmother didn't even think about what a virus is, and now she's aware of what a virus is, what it's capable of, and what she knows what a coronavirus family is. So it's also very interesting to see how people are getting more knowledge, like the general community is getting more a sense of what microbiology is and why is that important. Again, we also have diatoms and algae characteristics, and we'll go through them in the KoboToolBox. So where is all this information stored? And you can find this in the web page of the microdirectory. This is our current web page, but we are in the process of updating it. We hope to update it soon, and you'll have all the information from 2020 here. But basically what you can do here is to type any species that you want and get all the information that we have collected about that species. You can also download a set of microbes that you are interested on and do some research on them. You can also find us in GitHub. If you're familiar with GitHub, you know that you can download data, like huge data sets here, and code to analyze this. If you're not familiar with GitHub, I'll recommend you to just have a look at what we have here. We have a tutorial on how to download the data and what to do with that. So you can have fun and learn a little bit about GitHub. So the microdirectory, as you can see, is a database that requires a lot of work, right? We have volunteers that do the manual annotation, which is really important work because without this manual annotations, basically we wouldn't be able to make this data available for researchers and for everyone. We also have data analysts that do the data management, and we also have bioinformaticians that do the analysis of the biological data. So this is all the microdirectory team, and you can see here in the meeting that Krista, Ryon, and I are leading the microdirectory, but we also have some other members that are helping with the analysis and the processing all this data. So we are a pretty big team, and we're really happy to have you on board and to have you for you to be part in the team. So what will be basically your work in the microdirectory? I have pointed here 4 specific tasks, and we're going to go through each one of those and explain what they mean. And I know this is a lot of information, and I don't want you to worry about that. We will send you these slides over Slack. But if you have any questions, feel free to write them on the chat or unmute yourself, and we can have a discussion. I really like when you are interactive and talking and asking questions rather than me talking all the time. So let's start with the manual annotations. You will be assigned 10 microbial species each week. You have to make a constant weekly submission for about 12 weeks. So in total, you have submitted at the end of the cycle 120 microbes. The curation of the data means that you'll have to do some research on research articles and public databases to find the information that we required from that microbe. And the citations, it means for each answer that you find about that particular microbe, you have to reference that either article or database in the form that we will show you where you submit your data. And this is because we want to make sure, we want to give credit first to the people that have worked on in that microbe. And we also want to make sure that the data comes from somewhere and not just because someone, some individual says that's the answer. So we need to give evidence of where this information comes from. So let's start with manual annotations. As I said, you will get 10 microbial species each week. You'll get an email that looks like this, and it will say, here are your, and then it will give you the group that you will be annotating that week and a list of microorganisms. This list is sent every Monday. So if you have not received it on Monday night, please reach out to us through Slack because maybe your email is blocking our emails or something happened, and we want to make sure that you have enough time to annotate your microbes during the week. So if you reach out to us on, let's say on Thursday, that you didn't receive your microbes on Monday, then you'll have a really short time to annotate your microbes. And we have Krista is going to talk a little bit more about this, but we have a deadline, which is every Sunday night. So we don't want you to have, to accumulate all the microbes because it will be easier to annotate 10 than 20 at the same time, or 30. Are any questions so far? Are there any questions? All right. So the constant weekly submission, you'll be annotating these microbes for 12 weeks. And these annotations, meaning the submission of the data, is going to be performed through KoboToolBox. And Chris is going to explain what KoboToolBox is and how to do it. So don't worry. So in summary, KoboToolBox is a platform where you enter the data. And we have a YouTube channel where we show you tutorials on how to annotate each microbial group and how to submit it to KoboToolBox. So I'll highly recommend you to check this out before next Monday. Check it out and make sure that you understand and you have a sense of what you're gonna do and where to start, because we know that at the beginning, okay, you received your microbes and then you're like, okay, what do I do next? And we understand the first weeks are going to be a little difficult, but then you'll get the sense of how to perform the research and how to submit the data, so it'll be easier. But if you watch these videos, you'll have a better sense of what you're gonna do next week. We can grant extensions, meaning that, for example, that week you were really busy with finals, or you had a family issue or personal issue and you couldn't complete your microbes. Just email us to the email, the microdirectory at Gmail, and just tell us that you're going to be a little late in your annotations, and that's fine. Remember that we are quality driven and not just time driven. We want this to be as accurate as possible, because not only us, we're not only using the information, all other researchers are going to use it. So we want to give this really accurate data to every researcher, and even individuals that are not in research that want to learn about microbiology. The curation of the microbial data, you will be doing research in articles and public databases. We have a list of useful links for databases in the Coda doc. We're going to go through it. And we recommend you to search articles in Google Scholar or PubMed. And you can find information in other sources, but always verify that the information is reliable. We don't want really weird web page that you don't really know the source and that the information might be not really correct. So always make sure that the information is coming from a corroborated place, either article or database. All right, and the citations. We're going to be using so the citations are included in the KoboToolBox for each microbe. And we are using this website, which is SotheroBib, and it's very easy to find. You basically type SotheroBib and it comes out. And it's basically this blank bar, and you will paste the link of the article you want to cite. So, for example, I want to annotate a citation for this research article, this review on how bacteria recognize and respond to surface contact. So what I'll do is I copy the link of this web page of this journal and paste it here, and then you'll get something like this. So this is what you will copy and paste in KoboToolBox. In the last thing you do is paste in the citations. Again, you'll go through it in a second, so don't worry. But just wanted to make sure that you understand that we will be using the SotheroBib, and this is because we want to keep a standardization, like a standardized way on our citations. Are there any questions? All right. So the calendar for this cycle goes from February 22nd to May 10th, and that's 12 weeks in total. And we want to give you recognition for all the work that you do. And we have set these parameters for according to your work, the quality of your work and the time you're spending annotating this. And we want to give you acknowledgement if, for example, you only help 1 or 2 or 3 weeks and you couldn't continue. That's okay. We understand that perhaps the project is not a good fit for what you want, or you just didn't think you were going to spend so much time doing this and you really cannot do it. Just let us know through email that you want to withdraw from the project, so we know that you will not continue and we stop sending you microbes. But we'll give you acknowledgement in the publications, the upcoming publications that we publish in the future with the data you have collected. The people that continue with us will be part of the microdirectory consortium, and that means that the consortium is always a part of the co-authors in the publication, so you'll be part of the list of the consortium. And if you have been for almost the entire cycle, we want to give you the opportunity to work towards a co-authorship publication. And I know there is a lot of interest in here because some of you really want to get a publication to improve your applications for med school or for graduate school, and that's great, of course. And when we get the data that you will be annotating, we'll start processing it, and we'll let you know that okay, the data is ready to be analyzed, and if you would like to work doing like some analysis, some figures, or in the manuscript, you can help doing this and be part of the publication as a co-author. So we have a lot of resources for you to be in contact. We like to have like a really close contact with our volunteers. Not only okay, here is your list, you do your work. No, we don't want that. We want you to be part of our team, our group. So we have created different channels where you can contact us. And we have also some guidelines on how to do the work and how to do some troubleshooting. And this is the coda. And here you'll find, okay, what to do when I cannot find my species, what to do if I cannot find the information. We also have all the microbial definitions. Every single one of the questions we ask in Kobo are explained in detail in the Coda doc. So I really encourage you to read it all before next Monday. And we have some KoboToolBox instructions and some frequently asked questions. We have the Slack channel. If you haven't joined the Slack channel, please do so, because we share a lot of news or updates in the Slack. And I'll recommend you to download the app either on your computer or in your cell phone, because if you just open it in the browser, you will not get notifications of any message. And you can always find us there with direct messages. You can, for example, find me there and write me a direct message with a question, a particular question, or you can use the general channels where all the volunteers are active and asking questions. We also have the Dropbox. You can find the link of the Dropbox in the Coda, but in the Dropbox you can find some books and articles, and the glossary, which is like general common words that you will find in articles. Sometimes there are some tricky words that you really don't know what they mean, and they seem to be important. You can use the glossary. We have tried to make it as useful as possible for you. I really have to mention that some articles are not open. They charge you to read the article, and you do not have to pay for any article. If you need access to a specific article, let us know in the Slack, and we can give you access to it through Cornell or through other platforms. I think for some Hunter students, you have access through the institutional. Radwa, perhaps you could explain that a little bit, how it works.
Yeah, that's definitely a resource you guys have available to you. The Hunter Library database that's online that you can access with like your Hunter login, that has a lot of data, like, and a lot of kind of like the things you guys have on the Dropbox of just like online books of like microbial data that you guys can use as a resource in your annotations.
Great, thank you. So if you find an article that you definitely cannot find for free, let us know, put the link on the Slack and say, hey, I really need help on getting this article, and we'll help you. So you do not have to pay for anything. We have the YouTube channel. We also have, besides the tutorials, which are really great, I really recommend them. We also have some talks from researchers that joined us in the previous cycle. We'll host new talks this cycle. We will have some students, PhD students. We'll have some professors. So we will be working on the calendar for those talks. You're free to attend. You don't have to. It's not mandatory. But if you're really interested in a specific topic, you should attend these talks and learn a little bit more. And we have the Gmail. You can email us with extension requests or a personal matter or any more specific thing. Other questions about microbes, please use the Slack. Perhaps another volunteer has the same question, so feel free to use it. And all right, so I have talked about what like your work is going to be and what the microdirectory is, but what is the relevance of the microdirectory and your participation in the microdirectory? So first of all, you will improve your scientific reading comprehension. And it's not because Maria says so, it's because we have seen it with the experience of other volunteers. Even we have had volunteers that email us saying, hey, I really improved how just understanding this research article, or just like getting the information that I needed, which is something that you will use in basically any career in science. you will need to read scientific literature. So this is a great skill to improve, or to acquire if you don't have it yet. You will be contributing to an academic project and making this data accessible and searchable for everyone. The database can be used in basically any country, anywhere. And we actually have plans of like putting the microdirectory in other languages. We have actually the KoboToolBox in Spanish if someone is interested in doing it in Spanish. But in the future, we'll like to make it in different languages, so anyone that doesn't really know English can use it. We provided, as I said, this metadata, which is the data of the microbes, for metagenomic analysis. And that is to say, if you have a list of 100 microbes, you don't have to go one by one and annotate the information for one by one. That will take you forever. What if you have 1 million microbes? So this is why computational analysis are so important, because we can get the information only for the 1,000 microbes that I have, and the profile all these microbes in just seconds, basically in seconds. So you don't have to do it manually. You will be part of the microdirectory consortium, and this works as basically any like in the future, any paper, you will be part as a co-author in the consortium list. And this is great. We have another consortium for the project that Dr. Mason talked about, which is the MetaStuff project. We have collaborators from all around the world. And Krista can talk about that. She's the deputy director, and it's a great project where we not only learn about the urban microbiomes, but we also make so many collaborators that new projects are created with people that you will have never even meet outside the laboratory, or perhaps in a conference. But nowadays it's difficult to have this kind of event. And as I said, possibility to work towards a publication if you're interested on that. Whenever we are more advanced in the cycle, feel free to reach out to us and say, hey, I want to help with the analysis, and we can work on that. And listening to talks from researchers and professors. And as I said, if I think this is a win-win. Like, you get so much from us, but we also get so much for you. It's like we are a team, and we work together towards the same goal, which is creating this huge database. And finally, we have the contact that you can reach out to us anytime. Here was Dr. Christopher Mason, who is the principal investigator of the Mason Lab here at Weill Cornell. I am Maria Sierra, bioinformatician. Krista Ryon, who works with data management. She's in charge of the KoboToolBox survey, and she's going to explain it in detail. David Danko, who is a computational biologist, and Radwa and Hardik, who are volunteers that have been almost a year with us, and now they're helping to train and to guide other volunteers. So if you have any questions, email us to the microdirectory email, or if you have a particular question in the future, feel free to reach out to our emails. You can easily find our emails on Google. If you put our names and Weill Cornell, basically you'll find the directory of Weill Cornell and our emails. So with that, I'd like to take any questions, and I can go through CODA. All right.
So I think Maria's gonna go briefly through the CODA, and then afterwards I'll go through the KoboToolBox. So just hang with us there.
Yes. So you might be familiar now with the KoboToolBox. If you haven't checked it, please do so before next Monday. And the KoboToolBox is a document where we have all the information that you need to know to understand the microdirectory and to understand your microbes and the steps that you need to do to annotate these microbes. So in the first page, the microdirectory first page, you have all the links to the video tutorials, the glossary, the Dropbox, the web page, the GitHub, everything. And here we have the guidelines, what the microdirectory is. Well, again, what will be my tasks as a volunteer, where it's the KoboToolBox, important notes, everything you need to know. And please read this in detail. And if you have any questions, always it's better to reach out to us. And something important, if you email us and we haven't answered, you email us with a question regarding your submissions for that week, and we haven't answered yet, do not feel pressure to submit before next Sunday without actually making sure that you are completely sure of your answers. This is like a quality driven project, and don't feel like, oh my gosh, I need to send these microbes as how they are and that's it. We want you to be conscious about data and that you think the data is actually correct. So we have an explanation on what to do if you find an identified or unclassified species, and this might happen quite a bit, and that you get a species that doesn't really have a name. For example, this unidentified phage bacterium, and in this case, of course, you cannot profile this microbe because you don't know what this is. So in this case, you will annotate not NA, not applicable, to all the answers, and that is fine. You don't need to worry because we understand that this cannot be classified. If you are unsure what your microbe phylogeny or lineage is, you can check the NCBI taxonomy webpage, where you can basically type your species and then you will get the information that you need. I can go very quickly. You can write here your species, let's say E. coli. You will get E. coli and a list of all the strains of E. coli. I just need the species. And here you will get the lineage. So you know this is the Protobacteria, phylum Protobacteria, bacteria, phylum Protobacteria, class, order, family, genus. So you get a sense of what your microbe is. What do synonyms mean? If you cannot find the species information, what to do. Krista will go over this in a little bit. Annotating at genus level, you might get something like this, for example, Streptococcus sp. This means that this is a species, unclassified species, and from the genus Streptococcus. You can annotate this as long as the characteristic that you're annotating applies for all the species in the genus. So, for example, I give an example here. If the literature says that Streptococcus is a gram-positive genus, that all the members of Streptococcus are gram-positive, so then you can easily annotate this information. But if you find, for example, that in this genus there are some gram-positive, some gram-negative, you cannot put the gram staining. You cannot answer the gram staining in this case because that doesn't apply to all the species in the genus that you are annotating. In this case, you will put NA. So again, have a look at all of this, and after you have read all of this, feel free to reach out to us with any questions anytime. What is the difference between micro and macro? So I like to emphasize here for the algae and fungi. I'll go here. The algae and fungi, sometimes you might get a list of some fungi that are macrofungi, meaning, for example, this mushroom. In this case, you will not be annotating any characteristic of this. You will only select that this is a macrofungi, and that's it. The KoboToolBox will guide you through all the answers, and that's it. But if you are annotating microfungi, then we are interested in this. So you'll be annotating all the characteristics, if it's spore forming, if it's any antimicrobial resistance reported, and so on. So what is the difference between each one, the micro and the macro? Basically, the microfungi, you cannot, when you need a microscope to characterize all the parts, you will say, oh, but I can see the petri dish and I see here, this is the fungi. But you cannot characterize which is the spore, which part is the body, and which part is the head and everything. So you need a microscope to characterize it. By the other side, in the macrofungi, you don't need a microscope. You can see it with your naked eye. So basically, you know, this is the umbrella, this is the body, and the hyphae, everything you need to know. Only with your eye. So again, we're not interested in macroorganisms, we're only interested in microfungi. Same applies for algae. So there are some algae that are the ones, for example, that you see on the beach. You will see these are macroalgae, and we're interested in microorganisms. So these ones, for example, are examples of microorganisms. So again, have a look at all of this. We really have spent a lot of time trying to put all the answers to your questions. We have the KoboToolBox definitions. As I said, all the characteristics that we try to annotate for each organism, we have it here. What is, for example, for the virus, what is the genetic material, what does it mean, type of a strand, sense of a strand, capsid, and so on. We have really nice figures that we have taken the time to actually make them for you to make it easier to understand. We have some links to better understand the topic. And I like to stop here in the pathogen because there are many questions about this. And we only consider pathogen, that a species is pathogenic when the species, when this species, when present in the host, either human or animal or plant, when it infects the host, it changes the health of the host. So it makes it sick or develop some symptoms. And I say this because, for example, you will find some research articles that say that, for example, Streptococcus was found in an infection in an immunosuppressed patient. And we only consider pathogenic something that is not within the normal microbiota. So Streptococcus is usually in the microbiota of the airways, or sometimes the skin microbiota. So that's like a normal microbe. But whenever you're immunocompromised, everything will be harmful for you. So we only consider microbes that, when present, are bad for the host. So for example, the Ebola virus, you don't have Ebola just in your normal microbiota. If you get infected with Ebola, you will get sick and you will show symptoms. Same for example, for Zika virus or SARS. For example, some pathogenic viruses for plants, they get like this symptoms of the leaves, get like spots and things like that. So those are pathogenic, the ones that are not in the normal microbiota. And for bacteria, again, we have everything that you need to know. What the oxygen use, we have described each type of oxygen use, the metabolism. And here in the metabolism, this might be a little hard to find for some microorganisms. So don't worry if you cannot find a lot of information about this, because this characteristic in particular can only be described when the microbe has been isolated in the laboratory. So meaning that you got the sample, you put it in the petri dish, and you have your isolate of that particular microbe. And as you know, or you may know, that the majority of the microbes cannot be isolated. So only organisms that have been isolated and characterized will have information about the metabolism. We have described here some what they are. For example, what does it mean to be phototroph or organotroph or photoorganotroph, and so on. And we give some examples. So let us know if you have any questions with this. We understand it's a little bit tricky sometimes, but you will get the hang of it. Fungi, again, we have a additional question in fungi, which is if the fungi is a lichen. Right now, we're not interested in lichens, although they're really interesting. And if you're interested, actually, a little propaganda on myself. I have a paper here if you want to check it out with this really nice pictures of the lichens and what they are, the microbiome study that we did. But anyway, we are not interested right now for the microdirectory in lichens, but you might sometimes get a species of a lichen in your list, in your Wikily task. So whenever this happens, you will select the lichen option in the Kobo, and that's it. You will be done, basically. So read about what a lichen is, and so on. Same for microalgae. Again, as I said, we have spent a lot of time trying to characterize everything, to put like really nice references, really nice videos, pictures, the diatoms. So take your time, take some coffee and read it all, and let us know if you have any questions. And I will end with this. Also, we have the KoboToolBox instructions, but Krista will go through it, so I'll finish with these useful links. We have this list of databases that you can use to find information. It doesn't mean you will 100% get the information there. It means that at least the database tries to annotate it. Sometimes might not be present, but that's nobody's fault. It means that we'll just have to do some research on that microbe that hasn't been characterized. So try the second one and the third one and so on. So if we think this helps to actually have a narrow your options, and not just to find in Google, okay, where do I start? I have to find information in 2000 web page. You're gonna start with this. We have about 36, and all of them have great information. So I'd like to stop there to give the microphone to Krista. And again, feel free to unmute yourself to ask questions.
All right, thank you, Maria. Yeah, again, if you have questions, it's okay if you don't, but if you have questions, feel free to put them in the chat or just jump in and say something. I'm gonna head right into my PowerPoint just so we can wrap this up, and thank you everyone for hanging in there with us. I'm just going to go ahead and introduce myself again. I know probably said it like a few times over now, but my name's Krista, and I'm here with Maria, and Hardik's here now, one of our diligent volunteers for the Microbe Directory, who's been working with us closely, and he'll be one of the leaders for this cycle. So everybody say hi to Hardik. And yeah, as I said, I'm also part of the Mason Lab, and I do a few projects here. I work with specimens in the lab, and do a few projects as well as the MetaSub project, where we work on international sampling of air, wastewater surfaces, as well as some clinical samples. So yeah, I do a lot of work within the microbiome and microbes. So
We actually, sorry, just to add there,
and cats.
we actually last year had some volunteers from the Microbe Directory in the Global City Sampling Day. I think also Hardik was there. So if you want to join this year for the sampling, which is one day, Krista can explain a little bit more about that.
Yeah.
But we have also the opportunity for you to participate on that.
That's a great point, Maria. Yeah, so I do lead a lot of projects within the lab, especially those through MetaSub, and we do need a lot of volunteers to help us. Right now, the lab is not open in person for volunteers, but hopefully by the summer, we'll have some space for some new people to join us and help with some hands-on work. But as of now, I'll just go right into the Microbe Directory, which I've been a part of for, I'd say, yeah, about 2 years now. I joined in order to help with the data and to help manage the annotations. And I'll talk now about the Microbe Directory and KoboToolBox that you've been hearing so much about. And I'm sure you're like, what is Kobo? What is it? And I'll let you guys know. So KoboToolBox is a free open source tool for data collection. We won't be using it for mobile collection, but it is a platform that is designed for mobile and browser collection. It's completely free, compatible with most computer browsers. That is Firefox, Chrome, if you prefer Internet Explorer, and it's completely open source. It's designed for professional industries, humanitarian organizations, and researchers just like us. Since you guys will officially be researchers this coming cycle and be doing research with us. Can everybody hear me all right? I know it's a little loud here.
Yes, it's great.
All right, great. So KoboToolBox is really this data collection platform that's designed to help different walks of life collect data in a nice and concise way. And it has a few capabilities such as form builders and actually ability to analyze data. But we'll be focusing mostly on the data collection for the Microbe Directory. But I do have to say that if you do have time to kind of explore KoboToolBox and look kind of into it, that they have a big community, they have a GitHub, they do have a Twitter. If you have time to look into it or even make an account yourself, go ahead and look at it. I highly recommend it. It's kind of a big data collection platform, but it's certainly an interesting site. But you guys will be certainly familiar with it in the next coming weeks. You'll be on it quite often for the Microbe Directory. So this is what the form sort of looks like. This is what the form looks like when you log into KoboToolBox. We've been using this platform since last year. It's been almost a year now. And we've had a few different forms that we've made throughout the year, but this is the one you'll be using to enter your data, and so that we can track your data that you're collecting and entering it into our online server. So as Maria said, every Monday you'll be getting a list of microbes that you'll be looking at. You'll get 10 microbes, and they'll be sent every Monday, as I said, and your annotations are due every Sunday night by midnight. But also, as Maria said, it's not really on a strictly basis. So if you are a little concerned about your microbes, don't worry about getting it in on the dot. Of course, we prefer your microbes in before Monday, but just let us know. And what I want you guys to pay attention to is this right here. We're going to let you know what type of microbes you'll be annotating for that week, whether it's bacteria, virus, fungi, microalgae, or diatoms. Of course, you'll most likely be getting one of these first three, but you might run into a few of these algae. So do pay attention to what you are annotating. And you guys should have also received your KoboToolBox login and account that you'll be using, but I'll jump into that in a little bit. But yeah, make sure you're checking your email every Monday for this list. And this list is kind of what you'll be looking at every week for your 10 annotations. So the first thing you're going to look at is your domain, which is one of these. And then you're going to look at your list of microbes. And you're going to see, you're going to look through them, you're going to get familiar with them. And if you are ever unsure about the taxonomy of your species, so the full lineage, feel free to use NCBI. This is also laid out on the CODA if you're kind of unfamiliar with the site, but it is one of, it's a really great site for research. And if you're familiar with this sort of research, NCBI is a great tool to go on and look through. So I'll talk about it briefly here, but do go on the CODA and use NCBI taxonomy to get a better understanding of what your microbes are for the week. Okay, I'm gonna go right into your account and logging in. So everyone should have received their account user and login, as well as some info about KoboToolBox. If you haven't, I'm sure I'll be hearing from you soon. But what you're gonna wanna do to log in is actually press sign up, and it will scroll you down to what looks like on this page on the left. And you'll be clicking researchers, aid workers, and everyone else, because you guys will be researchers for these next 12 weeks with us. So you'll click here to log in, and then you'll see a page here on your right where you'll use your user and password that I provided to you to log in. If you do accidentally click the one on the left and try to log in on there, it just simply won't work. So make sure you do press the researchers, aid workers, and everyone else. There is a brief poll that just popped up, so just click if you did receive your credentials. Just give us a better idea of how many people got them. All right.
Hey, seems like everyone received their credentials. That's great.
That is awesome. Glad to hear that. But yeah, so that means you guys can log right in and do this yourself. So now you guys know where to log in, what to do. And once you log in, you should be seeing this page right here. And when you get on that page, you'll see the form that we have for this cycle. As of now, there's no submissions because no one's been assigned their microbes yet. But you, of course, can press on the form and make sure it says Volunteers Winter Spring 2021. You simply click right there in the middle, and it will open up to your own account page for the form, where you'll be seeing 3 tabs. These 3 tabs are the Summary tab, Form tab, and Data tab. What you'll be focusing on mostly is your form, but if you do want to browse around, your summary just contains a description of what the form is and the number of submissions that you've entered. And your data is actually what data you've submitted. So if you haven't submitted any data yet, that tab won't be there until you've actually submitted data. So you'll see that after the first week of your microbes. So what you'll be wanting to focus on mostly is your form. And once you get to your form page, looks just like this. You'll see a few things. You'll see the version model of the form, what languages it's in. Maria mentioned briefly that we have English and Spanish, so either or. And you can see this little eye on the top right where you could preview the form before you even open it. But what you're going to do in order to start your submission is just to click the open button on the bottom right. And just to note that each submission will only contribute to 1 microbe. So you'll be opening this form 10 times throughout your week to submit your microbes into the form. All right, so let's say you're ready to submit your first microbe. You've done some research on your microbe and you're getting into the form and you want to enter some information. So what you're going to do is you're going to enter your scientific name directly from your email that you received, copy and paste it right here in the text. And then you're going to look at your domain and click one of those, that bacteria, eukarya, or virus. And then enter your full name in the tab at the bottom. Depending on what domain you did receive, you're going to have a different subset of questions. Different questions will appear, so definitely put your domain in first before you start, or else you're not going to see the type of questions you need to fill out the form. To give you guys a better idea, so this is an example for viruses. So if you have a virus for that week, you'll click virus, and you'll see these set of questions that are based on the diagram that Maria showed in her PowerPoint. So you'll see the genetic materials of question, strand, capsid, symmetry, whether it's pathogenic. So definitely pay attention to Coda, go on Coda, look through it, because it's definitely going to translate directly into what you see in KoboToolBox and what you're going to need to answer. All right, so briefly, so let's say you're done answering all your questions and you're ready to submit data. You scroll to the bottom and you click submit, and you're going to see one of two things. You're either going to see that your record is queued for submission. That is great. That means your form is connected to our server, and uploading your data. And once it's submitted, you'll see this little yellow tab at the bottom that says one was successfully submitted. That is great. If you see the top one that says form contains error, it means that something went wrong. It means that maybe you didn't answer a question, maybe skipped it over. So just scroll back up and see if you see anything highlighted in red that might be wrong. If you do encounter a problem, you can always reach out to us, but it's usually that you maybe missed a question. Any questions that are mandatory are marked with a little star at the top, but I believe all the questions on the form are mandatory except for the last text-based question. So just make sure you're paying attention to when you're answering your questions. All right, so again, feel free to ask some questions in the chat if you're have some questions. I know this is a lot of information, and just a reminder that all of this is on Coda, so don't worry if you have questions or if this is a little confusing.
So there is one question in the chat. In the chat, where do we enter the citations?
That's a great question. So I don't have it here on the text form that I laid out in the PowerPoint, but from the form, you'll see a large text box at the bottom of your form. Right after you submit your name, there'll be a text base where you just copy and paste all of your citations into that large text box. It's kind of like these text box questions, but it's a larger paragraph form. Just a note, if you guys need some more clarification, I'll be holding a Zoom office hours at 9 a.m. tomorrow, Eastern Standard Time. So if you need more clarification or you just want someone to walk you directly through the form, I'll be on there from 9 to 10 to give you guys a little more time to ask questions or to go through the form directly. Or you could message me directly, and I can meet with you if you have any more questions. All right, so I'm going to go through these frequently asked questions quickly, and again, great questions. So if you have any more, just feel free to put in the chat. We're going to press on. So one question we get sometimes is Kobo keeps telling my username and password is incorrect. So as I said earlier, make sure you're logged into researchers, aid workers, and everyone else, and not humanitarian groups. The link has a KF in the beginning, so make sure you are on that and you should be able to log in. And again, if you do have any problems with logging in and that doesn't work, just reach out to us and we can help with that. Secondly, you go into the forms page and the main KoboToolBox page and you don't see anything. You don't see the winter spring form and you're not really sure what to do. Just email us with your username and that means your username doesn't have access to the form and we can give you access. All right, Kobo keeps asking me to log in after I submit an entry. So this happens sometimes when your browser has a cache problem. It means that you just need to close your browser and restart it. Sometimes it helps also to restart your computer, but you can also just use a different browser. It does just happen sometimes. So if you do have some problems with one browser, consider using Firefox or Google Chrome, depending on what type of computer you're using. All right, this is a very important one, I have to say. If you make a mistake and I didn't realize I submitted my data, can I edit my entry? Unfortunately, you will not be able to edit your entry after you submit your data. So make sure that you reach out to us and you put incorrect submission and your full name to our email and a description of your error. You could do one of two things right here. You could resubmit your entry with the correct information, And then you can let us know which one is incorrect, so we will delete the incorrect entry for you. Or you can email us with what is incorrect, and we will edit that entry. So pay attention to when you're submitting your data, and make sure it's correct before you put it in. But if you do make an error, just reach out to us and we can help you out. Right. All right. Near the end here. All right. I saved all my entries as drafts, and when I went to Kobo to submit my entries, they were all gone. So this cycle, we're not recommending that you use drafts. There is a drafts tab instead of submit on the form. We just recommend that you submit your data directly after you've completed the form. If you do end up saving your data in drafts, if you do prefer that, or if you want to explore that option, I would recommend just not closing your tabs and just making sure that, not closing your tabs, and make sure you're using the same browser every time you do annotations. Oh, right. And last question, what's the difference between NA and unknown in the data entry form? So almost every question on the form will have an option of unknown or NA, and sometimes you're going to come across a question where you're not really sure which one to put. So I'm just going to go through it briefly. So unknown kind of means not available or not known. More specifically, that you were looking for data on that said microbe, and it's just not there. It's the data you're looking for, it just doesn't exist. For example, if you're looking for the gram stain of a bacteria and you just can't find it anywhere within research, that would be unknown. It's just a simple fact that some microbes have more research and data than others. Some have been more research, and some have less research on it. So it's simply okay to put unknown, as long as you look to make sure that you did your best. And then NA and not applicable or not relevant means that the question it doesn't really apply to the microbe. So in this case, it's a little bit rare because Kobo kind of guides you into the questions that you need to answer for the microbe you're assigned. But sometimes you'll have to use this for microbes that, as Maria said, are, what's the word?
Unclassified.
Yes, thank you. Microbes that are unclassified. So none of that is going to apply. So pay attention if you do have an unclassified microbe.
Or for example, a genus level or a family level, that the question cannot be that characteristic cannot be described because it's so heterogeneous within that group. So you will put NA for that particular answer.
Exactly. So it can get a little bit, I wouldn't say confusing, but you have to just pay attention when you come across those answers and you're looking between what you need to put. So just pay close attention, and if you ever come upon a microbe and a question that you're really not sure whether it's unknown or NA, just reach out to us, either Slack or email, and we could definitely help you. All right, so Maria kind of went over it earlier, but here are some specific resources for KoboToolBox. We have our microbiology guides on Kobo, on Coda. So we have our definitions that Maria went through, and full instructions on KoboToolBox on Coda as well. So do check that out if you need a clarification or just a review of more. There's more information than what I said today. So yeah, go through that. And then if you have some specific problems, reach out to us through email for your account problems or incorrect responses, and we can help you there with some specific, more personal questions regarding your account. And then go on Slack for any questions related to Kobo or your annotations. We do have a specific KoboToolBox Slack channel if you want to join that, where you can discuss your problems or any other questions. I do highly recommend joining that group. And I would say just before next week, make sure you check out the Coda. It is just our dictionary for the microbe directory. browse through it, go through it before you start your annotations next week, and log into KoboToolBox if you haven't. Everyone has their user and login, so log in, check it out, open the form yourself, browse through it. There's no harm in just looking through it. And do join the Slack. We have over 300 people part of our Microbe directory Slack, which are our volunteers, collaborators, and it's a great place to connect with your peers in this time where everything is remote. And then do fill out the Google form I sent through email about KoboToolBox. It just lets me know if you were able to log in and you have access to the form. I believe most people have filled it out already, but if you haven't, do fill it out, and I'll be in touch if you have any problems with your account. All right, and finally, welcome to the Microbe Directory team. We really appreciate everything you guys do and all the work that you'll be doing for us these next cycles as researchers with us. And know that our cycle does start on the 22nd, this coming Monday. So be prepared, go through everything, and get excited about next week. All right, and I'll take any questions you guys might have.